I'm going to be going over the cell unit test study guide with you. Okay, it gives us an example here of a food molecule entering the animal cell and converted into energy. It says it enters through the cell membrane, goes into the cytoplasm, and there it's picked up by the ER where it is then packaged, goes to the nucleus, figure out where to go. It's packaged in a vesicle, goes out to the Golgi apparatus where it's modified and packaged and sorted and put into another transport vesicle and sent to the mitochondria. Number one, we're going to trace the path of a protein being made in the cell. And the protein is going to be made in a ribosome. And then from there, it will travel into the rough ER. That's the rough ER. Inside the rough ER, it will be folded and processed, and then it'll be put into a transport vesicle out of the ER. And that transport vesicle will take it over to the Golgi. The Golgi will modify, sort, and package that protein. And if it is an excretory protein, the Golgi apparatus, if it's an excretory protein, then it will be transported out of the cell. But the Golgi is going to modify, sort, package, and ship. It's going to package it into another vesicle. Package into a vesicle, and then that vesicle will take it, if it's an excretory protein, it's going to take it to the cell membrane to be excreted. If it's not one that needs to be excreted, then it will just be shipped off to whatever part of the cell it will need to be used in. But that would be the pathway made in the ribosome. Okay, it goes into the rough ER where it is processed and bent into shape. And then it pinches off at the end of the rough ER in a vesicle. It's picked up by the Golgi. The Golgi is going to modify it further. It's going to sort, decide where it needs to go, package it in another vesicle, and then ship it off where it needs to go. If it's an excretory protein, that'll be to the cell membrane to be excreted outside the cell. Number two, which organelle converts the chemical energy stored in food into usable energy? And that's going to be usable energy in the form of ATP. So we have food energy, like glucose coming in, and then it has to be changed into usable energy, ATP. So which organelle converts the chemical energy stored in food into usable energy? That is the mitochondria. The mighty mitochondria. And it's going to change it into ATP energy. We call that metabolic energy. What is the function of the cell membrane? Well, the cell membrane holds it all together, and it controls what moves in and out of the cell. So it controls what moves in and out of the cell. It also holds it together, holds the cell together. And there's special proteins that allow certain things to come in and out of the cell. Their osmosis and diffusion can happen across the cell membrane for some particles. It's controlling what moves in and out. We compared it to the gatekeeper. So here we're talking about prokaryotes and eukaryotes. Remember, pro means no. Prokaryotes are those cells who do not have a nucleus and do not have membrane-bound organelles. So U means they do. U is a true nucleus. Membrane-bound organelles such as the endoplasmic reticulum and the Golgi and all those. Okay, so number 4 says some cells have more mitochondria because the cells use more. Well, our keyword here, watch for those keywords. Our keyword here is going to be mitochondria. Here's our keyword. And the mitochondria does what? Well, the mitochondria makes ATP energy. So if they have more mitochondria, they can use more ATP energy. Use more energy in the form of ATP. It says mitochondria, we're going to think ATP energy. Number 5, a single prokaryotic cell can divide several times in an hour. Few eukaryotic cells can divide as quickly because eukaryotic cells are more blank than prokaryotic cells. Well, the eukaryotic cells have a nucleus and an ER and a Golgi and all those things that they have to copy. So they have to be able to copy all those. They have to make new organelles that are more structurally complex. So it takes them a lot longer to divide. So we're going to put structurally complex here. Their structure is much more complex, which means they have to make a lot more stuff before they're ready to divide. Structurally complex. I would look for that question to come up. So remember that term. They're more structurally complex. Eukaryotic cells are more structurally complex. They have to copy all those organelles, so it takes them a long time to be ready to divide. Number 6 is a prokaryotic cell can exist in a broad range of environmental conditions, can rapidly multiply, and lacks, well, pro means no. First pro means up. It says pro. Then that means it lacks a nucleus, does not have a nucleus. Does not have a nucleus. Animal cells do not have. Well, let's think of some things animal cells do not have that plant cells do have. Well, they do not have a cell wall. They do not have chloroplast. And they do not have a giant vacuole. There are a few small vacuoles in an animal cell, but they don't have the giant vacuole that the plant cell has. They do not have cell wall, chloroplast, or a giant vacuole. Not is our key word here. They do not have. So which organelle makes plant cells autotrophic? Well, autotrophic has to do with how they're getting food. Autotrophic means self-feeding. And that process is photosynthesis. And photosynthesis happens in the chloroplast. And the chloroplast is a plastid, and it contains the green pigment chlorophyll. And then chlorophyll is responsible for catching the sunlight, the energy from the sunlight that allows photosynthesis to take place. So the chloroplast allows plants to be autotrophs, allows them to do photosynthesis by capturing energy from the sun. Number 9. There's 3 parts that make up the nucleus. 3 parts that make up the nucleus. We have the nuclear membrane, sometimes called the nuclear envelope. It's the same thing, nuclear membrane or nuclear envelope. Then inside the nucleus we have the chromosomes. And the chromosomes are where the DNA is housed. They're attached to those chromosomes. And the chromosomes are attached to that DNA. So the chromosomes and the DNA, that's the genetic material that's inside the nucleus. And also inside the nucleus we have the nucleolus. The nucleolus is responsible for making ribosomes. So 3 parts to the nucleus: the nuclear membrane, the chromosomes that are inside, and the nucleolus that is inside. Chromosomes have the genetic material, and the nucleolus is making ribosomes. Cells with numerous ribosomes, there's our keyword again, ribosomes, are specialized for, well, ribosomes do what? Ribosomes make proteins. So cells with lots of ribosomes are specialized for protein synthesis. Synthesis means to make. So they're specialized for protein synthesis. Ribosomes, protein synthesis. Number 11. Passive transport differs from active transport because it does not require blank from ATP. Well, even if you're not sure what it doesn't require, if you see ATP, we know that ATP is what? ATP is energy. So passive transport differs from active transport because it does not require energy from ATP. Passive does not require energy. going to happen either way. It does not require energy. Endocytosis is an example of blank transport. Okay, endocytosis would be an example of active transport because it is requiring energy. The cell is going to wrap around and take in what it's consuming, take in, and that requires energy. So this is active transport. Number 13, blank is the process of maintaining constant internal environment. That's the word that we introduced back first or second week of school. And I said, you're probably going to hear that every single day for the rest of the semester. So homeostasis. Homeostasis, maintaining those internal conditions. That's keeping your body temperature around 98.6, your blood pressure just below 120/80. Your blood pH right around 7.1. Homeostasis is maintaining those conditions that are important for life to occur. Maintaining those constant internal environments. Even when the external environment changes, we go outside and it's 30 degrees, our body temperature should not drop to 30 degrees. We maintain homeostasis. Process that always involves the movement of materials from inside the cell to the outside of the cell is called, well, we're going outside. So we're moving to the outside. We are exiting the cell. That's our key word right there. To the outside. We're exiting. So that is exocytosis. Exocytosis. Exiting. Going out of the cell. Exocytosis. True or false? Active transport uses energy. Well, that is true. That's why it's active. You have to be active, use energy. Some examples of active transport. That would be endocytosis, exocytosis, and that can be phagocytosis or pinocytosis. Those are all active transports. Diffusion occurs because, remember, diffusion is passive, and it goes with the concentration gradients. As molecules are attracted to one another, molecules constantly move and collide with each other. Cellular energy forces molecules to collide with each other, or cellular energy pumps molecules across the cell membrane. But we're not using energy, so we know those are not true. So molecules are constantly moving and collide with each other. In diffusion, they want to even up. Nature wants to even out that concentration. So as they bump into each other, it spreads around and evens out. Okay? Which means a particle transport requires input of energy from the cell. So it's asking which one is using energy. So input of energy. Input of energy. Osmosis, diffusion, facilitated diffusion are all passive. Active transport uses energy. Okay, we're going to stop right here with number 18 for now. Part 2 will be tomorrow, and we will pick up with number 19. And that's it for today.